Good morning. This is our first video on the nervous system, and this video will be a little bit of an introduction. It'll kind of focus on a little bit of the big picture of the brain and possibly how it works a little bit, mainly just for interest. And then our next video will get into the electricity, like how is it that we actually have a thought? How does a nerve cell send a message along its axon? That's the long skinny part of a nerve cell. And then talk to another nerve cell with neurotransmitters. Throughout the videos, we'll also get into drug addiction and things like that, and how psychoactive drugs work, whether they're illegal or legal, prescribed by a psychiatrist, or something, that's illicit off the street. So to start off with, there's this, and you can't get sound. Again, my setup is not allowing me because this is a pretty old monitor. But if you wanted to Google it, it's kayak. And essentially what's happening is there's a neurosurgeon back there, and he's moving some instruments around in this guy's skull, and then he's typing. And I think this was a halftime Super Bowl commercial, but Kayak's like a travel website, kind of like Travelocity or something like that. And so he's basically saying, hey, I'm busy. I don't have time to search websites for flights and hotels. So of course you can Google it, and the dialogue is actually pretty good. So as far as how realistic is this, of course, it's not 100% realistic, but there is kind of a little bit of truth to it. So it turns out in your brain, let me scroll down here into the 70s, so many pictures, we won't get to see them all, but we'll get to see some of them. It turns out that our brain, in general, does have some areas that are dedicated to certain tasks. So some types of things, like memories, are kind of spread everywhere. There isn't like one spot in your brain where you store memories. There are certain spots in your brain, like the hippocampus, where you convert short-term into long-term memories. But memories that deal with vision are stored in the visual cortex, which is back here. This would be what my brain would look like if I was turned. I don't know why I spun around, but if I was turned this way, this is what it would look like. So your visual cortex is back here. Hearing is right here. So if I have a memory of, my favorite song, and you were, scanning my brain, and they told me like, hey, think about your favorite song, or I was just listening to them tell me things, that part of my brain would be lighting up. What we can do is with scans, we can see where is sugar going, or where is activity happening. And so we can sort of see which parts of your brain are working. And then if I said, picture this or picture that, or think of a memory of, a loved one's face or something like that, then the visual part of my brain would light up. So again, I might have a specific spot, we think, for converting short into long-term memories, but I kind of store them everywhere. So there are some specialized areas, like in most people, speech is exclusively on the left side of the brain. So I have two speech areas, for example. One of them is called Broca's area, right there, named after the scientist. And another one is called Wernicke's area, named after the scientist. And Broca's area is the part of your brain that deals with making your mouth physically do the correct movements. So, for example, as I'm talking right now, I'm moving air across my vocal cords in a certain way. I'm tightening my vocal cords in a certain way so I can go higher pitch or lower pitch, so that I don't have a monotone voice. And then I'm like moving my tongue in different ways to make like a s sound or a t sound. For R's, I didn't grow up speaking Spanish, so I can't roll my R's correctly. Rojo. I know that sounded authentic, but I can't roll my R's properly. And that's a problem with my Broca's area. So if I had grown up learning to roll my R's, I could maybe sound more like a native speaker whenever I said Rojo. And that's down to my Broca's area. If you want to think of it this way, Broca's area could be, it's not the only explanation, but if I had a stuttering problem, pretend that I always got stuck on my S's or something like that, that's a Broca's area issue. Wernicke's area, which is located just a little bit more like, it's on the left-hand side. Spin around again, I don't know why. Wernicke's area is more back here, whereas Broca's is more in the front. Wernicke's area is where you actually comprehend speech, where you understand speech. So that's like, nouns and verbs, past tense, present tense, vocab. So that's understanding the language. So it's possible that if my Broca's area is damaged, that I can't get the words out. It's very laborious for me to just say a sentence like, I need to go out to my car, but I can't find my keys. But my Wernicke's area might totally understand that. So if you're talking to me, I'm downloading and understanding everything that you're saying, but I just can't get it out. And I know that I can't get it out. So just like somebody that has a stutter, they are fully aware that like they're stuck on those letters, but their Broca's area is just not working well. The way that we figured this stuff out back in the day, because we've known for a long time that this was Broca's area and that's Wernicke's area, is you'll generally have somebody, let's say that this is a brain, so pretty close resemblance. And let's say that you have somebody, their speech is normal, and then they get in some sort of accident, some sort of farming accident, whatever, heavy machinery, car wreck. And so now this person that spoke completely normally before, it seems like the only thing different about them after they recover from their injury is now they can't speak. It takes a lot of energy. And so what we would do is after that person dies, when we do an autopsy, we'll notice that that area is damaged. Okay, well now you have another patient and another and another. And if they all keep kind of having similar symptoms, and then after death we realize they have damage to the same part of the brain, we're like, that's a speech area. This one over here, we figure out that whenever it's damaged, you, again, can't understand language. So here's what's interesting. Let's say that I go in for surgery, again, kind of like the kayak commercial. Let's say that I go in for brain surgery because there's a tumor down deep in my brain. So let's say that this is my brain again. I'll make that the frontal lobe. And down in the middle of this, pow, pow, pow. So right in the center of this cylinder, this brain, is a tumor. And the only way that you're going to live is if we cut it out. So I've got a few options. I could cut through here, or here, or here. I've got several different angles, but I'm going to have to destroy some of your brain to get to it. And what we know is, in general, brain cells are not very good at recovering. So whatever brain tissue we destroy, we're destroying that. And if that particular area that we destroy happens to control speech of some type, like Broca's area, if we drill through your Broca's area, like you're not going to speak normally again. Or if we drill through your Wernicke's area, you're not going to be able to comprehend language, whether you're, input or output, understanding or kicking out. So we want to make sure that your brain is actually mapped like this. Again, this is a rough outline of what most brains look like. Not everybody has speech on this side of their brain. Some people it's on the opposite side. And that's not necessarily linked with whether you're right-handed or left-handed. We still don't know for sure why about 90% of the population is right-handed and about 10% is left. So, but speech could be on the other side. So what we don't want to do is go into surgery, think that we're cutting through this part of your brain that controls, maybe like your right pinky, I guess that'd be the left side of your brain, thinking that we're controlling your right pinky, and then find out that we removed your ability to speak or something. So during surgery, back to the commercial, you're actually awake. Now this guy is not, but in real surgery, you are. So they'll numb your skull, numb your scalp, and everything, and they drill through there, but your brain itself doesn't have pain receptors. Your brain can sense pain, like if I smash my hand, my brain is the one that feels that sensation of pain. But my brain itself, if you were poking my brain, it doesn't hurt. So we numb the area, but then you're awake. So it's kind of like a dentist. We can numb the area with lidocaine and novocaine and stuff, and then we can drill on your tooth, and that part's numb, but you're still awake, like you're still watching, whatever home show they have on at the dentist office. So here, he would be awake. And what they do is they basically start mapping out your brain. And so they'll go in and shock little bitty areas of your brain, and your brain will respond. Your brain runs on electricity. So as far as the sensation goes, if I can feel this, like I can feel my finger touching the board lightly, and then more heavily, and then harder and harder and harder, that is because I'm sending electricity to my brain. And when that part of my brain lights up, it says, feel this, and I feel light pressure. If I hit harder, then more nerve cells fire, And so now my brain says, oh, feel this, and it's like heavier pressure. Okay, so what we do is we shock your brain, and then you tell us what you sense. So if all of a sudden area H right there controls maybe my right pinky, then when we shock it, if it's motor control of my right pinky, then it'll move. If it was sensory control and we're shocking it, I'd be like, it feels like you're touching my pinky. If we hit a speech area, if we were to hit your Broca's area, then you'll either have great difficulty talking, or you would speak just kind of, you know, some gobbledygook, some gibberish. So you might be like, hey, keep talking, tell us about what you did yesterday. And then whenever the electrode touches this, that's going to represent it getting my Broca's area. I was like, well, yesterday I went to the store, and then after that I went and got my car worked on, and then because it's a speech area, it might just mean that I kind of speak some gibberish, but I will somehow have an outcome with my speech. And so we map your brain, and they basically just put little markers on there, because eventually when we're going to drill through and get to that tumor, we're going to destroy part of your brain. So we map out my brain and we're like, we can drill here, that's a great solution. Oh wait, you'll no longer be able to speak. Or we can drill here, that's an equally good angle, and you'll lose the ability to control the last inch of your pinky. You're like, all right, who needs a pinky anyway? So in surgery, this is what it actually looks like. Oh, by the way, this picture will be a little bit rough. So right here, they've got everything pulled back. All these strings are just holding things back. And I know this looks fake, but it's not. That's an actual brain in surgery, and they've got little bitty markers on there. So somewhere over here, somebody's got a little clipboard, and it's like, hey, what was area C? What was area B? What was area 10? That type of thing. And they've mapped out his brain before they drill through to get to the tumor. Kind of crazy if you think about it. It's also weird to just think that if you were shocking the part of my brain where I store my memory of the smell of like pizza, then as you shock it, like I would be smelling pizza. So they shock it and they're like, what do you notice? And you're like, smells like pepperoni pizza, you know, or smells like grandma's house or something like that. So it's pretty bizarre. The next few pictures I'm going to show you are all kind of like real pictures of cadavers. A cadaver is a dead person. You might have dissected an animal in the past. When you dissect a human, we say that you're dissecting a cadaver. I'm not sure why we name humans unless it's something out of respect. So the next few pictures will be a little bit gross, but not too bad. So this one right here is showing that the brain has these covers called meninges. And there's 3 layers of meninges. You don't have to know them all. But these layers of meninges kind of like fasten the brain down to the skull. So if you think about it, here's my skull, and my brain sort of sits in there. Well, if I do a handstand, doesn't my brain just now like fall in at the top of my skull? Well, it doesn't, because I basically have these meninges fastening it down. So here's my skull, here's my brain. I just sort of have, you can think of it as functioning like bungee cords, but it's kind of like little sheets, almost like canvas. And again, I've got like these 3 layers of sheets. I have little sheets that fasten it down, and then another, and then another. And so that's what they're peeling back right there. Those are the meninges. And whenever the meninges are inflamed, itis means inflamed, you have meningitis. And usually inflammation is caused by infection, virus, bacteria. And so that means that if you have meningitis, you have a bacterial or a viral infection, most likely, on your brain or spinal cord. And that's bad, right? It's one thing having an infection in your arm, give you some antibiotics, take care of it. If some cells die, no big deal. But now if I have bacteria or viruses growing on my brain or my spinal cord, when you start damaging brain and spinal cord cells, again, the rule is they really suck at growing back. So that tends to be like permanent damage. So meningitis is bad. Those of y'all that are heading off to college, before you can go to college or at a minimum get into the dorms, they make you get a meningitis vaccine. So when you're 17, 18 and all that, and you're applying to colleges, again, especially if you're going to like live on campus in the dorms, then they'll require you to have a meningitis vaccine. And again, different states might be different, but I know here in Texas, that's the rule. By the way, one quick aside, For today's lesson, you don't really need any notes or anything, but in the future, there is a set of notes. It's called PreAP Biology Nervous System Notes that look like that. And the place that you can find them, I'll show you really quick, is if I go to our Canvas page right here, and I go off to the side. I know you can't see this very well, but if I go off to the side and click Files, then it'll show up in there. It says PreAP Biology Notes Body System. So click that, and there you go. Again, for this video, we're just kind of introing interesting brain stuff. You don't need the notes. So back to some cadaver images. Here's a picture of a human brain and spinal cord removed from the body, which is pretty difficult to do. So I had that privilege. Let me grab something. I had the privilege of dissecting cadavers in college. My human anatomy class, we had a male and a female cadaver. I didn't have one all by myself, so we dissected them as groups. But one of the things that I found was interesting about like the human brain is the version whenever you're going to make a cadaver, you take out all the body fluids and you replace it with preservative. So whenever you were to cut on a cadaver, you don't have blood come out, it's preservative. That way the body doesn't rot, which I know seems gross. Well, the preserved brain, this will represent the brain, is actually pretty fragile. And a fresh brain is also pretty fragile, but the preserved brain is a little bit more firm, from what I understand. And the thing that was surprising about it, and I've held probably, I don't know, 6 or 8, maybe 10 human brains in my hand in my lifetime. But what's weird about it is it's about as fragile as like room temperature Velveeta cheese. Just a little bit more firm than that. And so what's interesting is like if I take this and it was room temperature Velveeta cheese, and I just kind of like, you know, hit it on this counter over here, it got a dent. So it's surprising that we aren't more messed up than we are because it's so easy to damage brain tissue. But again, those meninges kind of fasten down my brain to my skull so that if I'm boxing or playing football or MMA or whatever, my skull is here, my brain is here. There's not much distance between my brain and my skull, but it's kind of fastened down. So when I'm running and all of a sudden I stop because I collide with somebody, hopefully my brain doesn't like smack into my skull. If it does, that's a concussion. Whenever your brain is bruised, or it can cause a concussion, that's brain bruise is a concussion. Another picture of a cadaver that I want to show you. Let me scroll into the 70s here. This one's a little bit rougher, but this is a guy looking to his left this way, and then they just cut straight down the middle like this. So they cut this way. And you can even see, if I zoom in a little bit, you can see right there is his scalp. That outer layer is like the skin. That second layer in there is the bone. And then underneath that, you can see some of the meninges. So the brain doesn't normally look like that. That's the brain covered with meninges. And we just cut like right down the middle. Here's a view head on. So this is, if I look right here and they just cut this way, like, you know, like that, right through the middle. And on this guy, you can even see he's kind of got that old man, real close kind of buzz cut. So he's got a little bit of hair there. That's his scalp, that's the skull, and this is the brain. This is, from what I can tell, I'm not a, you know, a neuroscientist or a neurosurgeon, but this is a normal looking brain in the sense that like those holes in there, those ventricles are supposed to be there. Now, somebody else with more knowledge than me might say like, hey, that little spot right there, or that spot right there, that gray area should be bigger or smaller or something like that. So that I don't know. But that kind of butterfly looking, those openings, those ventricles, kind of like the ventricles in the bottom half of your heart, are supposed to be there. So it's interesting. Many things are interesting in this picture, but the space between the skull and the brain is pretty small. So that space is filled with CSF, cerebrospinal fluid. It surrounds the cerebro, the brain, and the spine, and it's a fluid. There's also cerebrospinal fluid in those ventricles too. But again, it's surprising that my brain is really close to the skull. So when I get punched or I collide with something or I fall down the stairs, it's kind of surprising that my brain doesn't ram into my skull more often. Another thing that's interesting is when your brain swells, it basically smashes itself. if my arm swells, let's say I break my wrist, my arm is going to swell and swell and swell and swell. Well, it can technically expand into like, you know, infinite space almost, like the whole room. It could in theory, now this isn't gonna work, but it could in theory expand and expand and expand and expand. My brain can't, because it'll start smashing itself against my skull. So when your brain swells, it can just swell a little bit before you start having pretty major problems. And what's kind of odd is where your brain exits your skull, the foramen magnum, big hole, down here, that's kind of the only exit. So if fluid starts building up around my brain, I can't expand my skull because that's bone, so it starts pushing my brain down through that hole. So down in the base of my skull, I have the foramen magnum, and that's where like my spinal cord goes. Well, now you're just sort of like shoving your brain through that hole. So picture you have like the squirtable grape jelly or whatever, and you're just kind of like pushing it out, and it's just getting wrecked as it goes. So that's why brain swelling is a problem. Here's another picture. This is a brain of somebody turned and looking this way, and they sliced right down here. So right between the eyes. This is a very clean looking brain. And in this one, you can see the cerebrum. That's all the stuff that looks kind of like worms. And you can see the cerebellum. That looks like cauliflower cut. What's interesting about this picture is all of this stuff that looks kind of like worms, the cerebrum, my left and my right half of my brain are largely unconnected. They are connected, but they're largely unconnected. So it's kind of like this. Here's my right brain, here's my left brain. A lot of it is two halves just sitting next to each other. Like I could literally put a piece of paper between those parts. That's what's happening right here. If I had the other half of the brain right here, I could slide a piece of paper once the meninges were gone and go all the way down until I get to this kind of thing right here. The corpus callosum. The corpus callosum is what connects the left and right half of your brain. So from here down, basically everything that I'm covering up is connected. Everything that's not covered up, that right half and that left half aren't connected. So the right half and the left half talk to each other through the corpus callosum. You might have heard of corpus callosum also. I'll even probably switch saying it different ways. So the corpus callosum is interesting because that's how my right and my left half of my brain communicate with each other. And the reason that matters is because in a future video, that means like, for example, speech is on one side of my brain and most of us left, and writing is on the other. My left hand, information from my left hand goes to my right brain. Information from my right hand goes to my left brain. So if my corpus callosum is severed, we do this on purpose in some epileptic patients. Again, we'll explain this more in the future. But if my corpus callosum's severed, then all of a sudden, like, if I touch something that's familiar, like, you know, this, I'm a teacher, I've been holding Expo pens for decades, so if you put this in my hand, I'll be like, oh yeah, that's an Expo pen. Yeah, I can even smell it, it's an Expo pen. So if you put this in my left hand, and then like ask me to tell you verbally what's in my left hand, this went to my right brain, but speech is on my left brain. So only my right brain received the information. My left brain is in the dark, so I'm like, I don't know what it is. But then, if you give me a pen and a piece of paper, and my right brain controls my left hand, and you say, write about it with my right hand, I can't. But then, if you say, okay, write about it with your left hand, I can be like, oh, now I'm a lefty, so you can't read it very well, but oh, that was an Expo pen. So it gets kind of interesting, and we'll explain that in a little bit more detail just a little bit later on. I wanted to show you one last picture of the brain. This one's a fresh brain, so this one's probably the most gruesome. This would be if you were at like an autopsy, you cut somebody's brain out and you just set it down, upside down, and take a picture of it. So again, the reason this one's kind of gross is it's fresh enough that you can even see like little bitty droplets of blood coming off of it. And the thing that I'm wanting to show you there is the natural color of the brain. Most of the other brains we've been looking at are preserved, and so the coloring is not typical of what you would see in an actual living organism. As you can kind of see how it has this grayish pinkish look. we'll say it has gray matter, but then it's covered in blood vessels that are red, and so it gives it kind of a grayish, pinkish sort of hue. If I cut through a preserved brain, let me pick a different one. Right there. If I cut through a preserved brain, you can see that there's gray matter and white matter. So around the outside, this is like if we were making slices, almost like slicing bread, like a loaf of brain, you know, a loaf of bread, and we're just like, and we pull a slice out of the middle. That doesn't sound crazy at all, does it? So here we can see the outside edges are gray. The middle part is white, and we'll actually explain why that is in the next video. So thanks for the brain intro, or listening to the brain intro. And our next video will focus on how nerve cells talk to each other, and we'll keep relating that back to like, how does the brain work? So we will get into like the small details of how we think, but we'll also keep relating it back to the big details of like, how does that look at the brain level, not just the cell level. But anyway, we get to think about thinking.